Okay, let's have a class, and we will move to our last section of this whole AEM 341. The outline of exam 4 we will talk about is applied microbiology. First of all, we will talk about the food microbial safety today. Then, after Thanksgiving holiday on Tuesday, we are going to talk about poultry meat microbial safety. Because we have done tons of research for that, and there's a lot of topics we could talk about. Then following Thursday, we will talk a little bit about immunology. Followed by that, we will mention briefly about virus and also the final exam review. So this is pretty much outline what we're going to do. Right after Thanksgiving holiday, after next week, I will post it on the eCampus about the bonus points of the final exam essay writing. Okay, because it's a little earlier, I would, by the time after holidays, you enjoy your holiday first, and after holidays, we're going to post it on the eCampus, talk about that. Okay, so that is a plan, how we're going to do. So today the topic is microbial food safety in the United States. So we want to talk about the whole situation, the pathogen related to that. Some of them we already mentioned in the lab or in the lecture here and there, but we're going to be more focused to talk about today. So first of all, this is surveillance results from CDC. Now, by the way, what's the full name for CDC? That is Centers for Disease Control and Prevention. In 2010, they did a surveillance survey in the United States. What we find, we have 48 million cases of foodborne illness every year. 120,000 people will be going to hospital because of foodborne illness, and the results 3,000 deaths. They suppose to have a new data come out in 2020 because of the pandemic, so it's delayed. I think it will come out sometime soon, or maybe 2030. So we'll see. But this data is a little older, but we can see the number is huge. So in the United States, to be honest, in the history, at the very beginning, nobody cares microbial safety. So we know E. coli, O157H7, for example, it's a very common, you heard about every day, the foodborne pathogen. Nobody cares. The first case comes out is 1982 in Oregon, in a cattle plant. And here and there have Listeria salmonella, and by the time in 1980s, people doing more research is a nitrite to control endospore-forming bacteria like Clostridium. Today's food safety, all the regulation, all the research and extension, all comes out these outbreaks. That is 1993 in Jack in Box, Seattle. Because of customers eat undercooked hamburger with ground beef in the middle, it caused a damage, a huge damage. The undercooked hamburger is contaminated with E. coli O157H7. It results 171 people hospitalization. Most of them are 10 years younger. That is a risk group of foodborne pathogen. Young people, elderly people, immune-compromised patient is high-risk group for all foodborne pathogen. 45 infected children required hospitalization. 28 years suffered serious kidney problems. But what's the problem? Really touch the nerves of everybody is 4 kids less than 4 years old are being died. This is what touched the nerve of the White House. So that's why by the time the president is Bill Clinton and the Vice President Al Gore, they work with the Congress, come out with the regulation. This is what happens. The hassle. So that's a history. Now we want to talk about this pathogen first. I talk about the whole semester. So E. coli 0157H7. What this means? E. coli 0157 is Escherichia coli. Genonym, species name. This is the antigen. And this is modified sugar. Or we call it ceramic. And this is flagella. Serological typing. (157) and H7. These pathogens specifically will cause Shigella toxin. Shigella toxin has Shigella toxin 1 and Shigella toxin 2. Most of the time, cause of problem is Shigella toxin 2, and their target to the kidney. Because in the kidney there is a complex called GB2, GB3. They could firmly attach to it and will cause the disease. HUS. Hemolytic uremic syndrome, which is blood kidney with water diarrhea. And this pathogen is also very invasive. It could be going to the spinal cord, which is cause neurological problems, and eventually will cause people death. And the young kids is the most most affected, vulnerable group, we say. So today, now by the way, this is also AB toxin, okay, like we did before. So today, the regulation regarding E. coli 0157 on ground beef is 0 tolerance. What this means, if you get a bag of the ground beef from Kruger, it should not have 1 cell of 0157H7. It's not 1 cloning, it's 1 cell. If there is 1 cell tested a bagged ground beef, we call it adulterant. That's a strict regulation. However, you heard about today, there are lots of the news comes out with E. coli 0157H7, recalls and outbreaks everywhere. It is difficult to control. Okay? Now, regarding the ground beef, USDA, FSIS, Food Safety Inspection Service have guidelines. You need to cook the internal temperature of a beef products to 160 degrees Fahrenheit, which is 71.1 degrees Celsius. This is a tricky thing is here. This is the beef. What this regulation, what this guideline is talking about is the internal geometric temperature is above 1.1 degrees Fahrenheit, 71.1 degrees Celsius, 160, sorry, 71.1 degrees Celsius. this temperature will be effectively to kill E. coli 0157H7. Now, why ground beef or beef steaks has this issue? All the ground beef, beef steaks, or mechanical or chemical tenderized beef, originally from the carcasses, let's say this is a beef carcass, it should be no pathogen. And our tissue, like the muscle tissue, should be sterilized. But when you cut it, you ground it, you touch all the utensils and all the processing facility, it's easily to contaminate it with 0157H7. And originally that's coming from the soil, it's coming from animal fecal samples. So they get contaminated and could be penetrated into the muscle structure once further become a beef product. So all these kind of beef products, we all call those non-intact beef. And the regulation is the internal temperature. It's not on the surface, because the surface temperature usually it goes up very quickly. The thicknesses there are going to affecting the transfer of the heat. So remember the heat, we do conduction, transformation, and the radiation. Remember the physics, we talk about those three methods for the heat to transfer. In the conventional oven, usually it's a convection or conduction transmitter for the heat. So that's a thing you have to know. Now, how are we going to make sure 71.1 degrees Celsius? You need to put a thermometer, which is insert into the geometrical center. Now, the question will be, why there is an issue for this? The reason is beef products, we talk about tenderness, we talk about juiciness, and we talk about flavor. This is we talking about is palatability of the beef. Lots of people like the tenderness. If your tenderness increased about 10%, they are willing to pay even $5 extra for one small piece of the beef. Because tenderness is a more favorable factors will be considering by the consumers when they buy it. Now, one of the reasons to do the tenderness is you cook them to medium well done or even medium status. So, this is the issue for the beef. There's a problem. So how are you going to find the balance about that? It is tough. Back in 20 years ago, when I was a PhD student, we did a ton of the research about the cooking studies to figure out the temperature using different utensils, different appliances, how long we're going to cook to reach the internal temperature of 71.1 degrees Celsius. But it's tough because when you cook that well done, the beef will not be very tender. So this is something I want to mention. The reason why it is tough because of the tenderness, people are thinking about that. Now in the chicken products, there's no that concern. Because chicken, we don't eat raw. The reason is the muscle structure of beef and chicken are different. Okay, so that's something I wanted to say. Now, because of these outbreaks, Congress wants to do something. So they introduce a proactive preventive control system in 1994. That is called Hazard Analysis Critical Control Points. The full name is HACCP. This is a proactive control. Originally is coming from the space, coming from the Navy, the Air Force. Because if you are going to the space, you absolutely need to make sure your food is zero tolerance for the pathogen. So they're using this system to do. And this is covered honestly from the ingredients, the processing, the handling, storage, packaging, and distribution. So you can say this is covered most likely during the whole food processing. And what it is about, it has 7 principles. and it becomes a law in 1996, published as Federal Register by Department of Agriculture Food Safety Inspection Service. So the 7 principles is required by every meat and poultry producer in the United States. You must have a plan for your meat processing. Now, based on their scale, the large establishments or large company needs to have it immediately in 1997. The median size allowed to have 2 to 3 years delay, and small and very small localized producer allow you more time. I think it's about 22,000. They should have well established those systems. How they do it? Because lots of those producers are with only high school diploma. They're not well educated. So how they do? This is rely on the 8062 Land Grant University, the extension specialist, to help them do that. So I had like 30% appointment of the extension with the extension service. Sometimes I do that. But we don't do here too often. The best place you should look for, if you want to get a certification by yourself, is Pennsylvania State University in College Park, about 3 hours away from here. Their extension program is really good. Have the short course and the extension training for all the 7 principles of the HACCP. Basically, we call it a HACCP training for meat and poultry products. Now, what are the 7 principles we talk about? That 7 principles is written down, easily to do, easily understand, but it is difficult to do. Here, we're going to introduce you briefly, because this could be a separate class teaching a whole semester. So the 7 principles of HACCP is number 1, conduct a hazard analysis. I just write the acronym. What is a hazard means? Biological, physical, and chemical. Chemical, let's say mercury, it's a toxin. Physical, let's say debris, dusts, metals, those are the risk. But honestly, in this hazard plan, we are more concerned is biological. That is bacteria, virus, and parasite could be. So, when you have a product, you need to see which hazards is reasonably will be happen, or most likely will happen. Then, you need to draw the whole food processing outline and find out, determine critical control points, which means during the food processing, there is a step which intervention could be applied to minimize the pathogen below the regulation. So for example, if you are cooking a steak, you are preparing a steak, the cooking processing, this is CCP, that's critical control points. Because that is the step will minimize or reduce the pathogen following the regulation. Here the regulation certainly is 0 tolerance, which means after cooking, ideally you should not find 1 cell of E. coli or 157H7. So in order to do that, we need to establish a critical limits. This is a physical and chemical parameter corresponding to the critical control points. Here, the critical limits is 71.1 degrees Celsius. Internal temperature 71.1 degrees Celsius. In the HACCP regulation, the critical limits cannot be a range. It has to be a fixed number, which means I cannot say my critical limits is 70 to 74 degrees Celsius. That is wrong. In the new regulation of FSMA, for the Safety Modernization Act, under preventive control, human processed consumed foods, it allows you to have a range. If you talk about HACCP, it's not allow you to have a range, cannot be a range. So when you have a hazard analysis, you have a critical control point, you have a critical limits. What you're going to do? You're going to have a monitoring procedure. So what it means, a monitoring procedure? Which means during the cooking process, I need somebody there to make sure you cooked it. The internal temperature is 71.1 degrees Celsius. That is called a monitoring procedure. That could be done by another person there. That is called quality control. Or it could be done is monitoring the thermometer temperature. So that's called a monitoring procedure. Now, in the real food processing, more or less, you will make some errors. Okay, you're going to make some errors. So, you have to establish corrective actions. So, for example, if one person is cooking 71.1 degrees Celsius, and you find that after cook 10 samples, you find that the temperature is not reaching that. That is called a deviation, which is failed to meet the requirements. What you should do? You need to take corrective actions. You could be taking them to recook, or this whole batch cannot be going to the markets, or you further cook to well-done products, which means the beef making a hot dog, because hot dog is ready to eat, has to be well cooked, or you make it as a pet foods. So there is several things you have to do. It's called establish critical actions. Now, once everything is working, we need to establish a verification and validation. What this means, which means we need to have an in-house quality assurance person to make sure you're following the protocol. And we also need a third-party audits to come into your plants to see you really did it. So that's called verification and validation. Finally, how do we know you did a good job of validation and verification? You need to do a good job of record keeping and documentation. This relies on the easiest one for this temperature. Obviously, is temperature data log, or we say data sheet. Every temperature, every steaks during the processing line, during the cooking, you measured and recorded the temperature, the internal temperature, whether they reach 71.1 degrees Celsius. So that is the 7 principles, what they are looks like. It's easy to say, but difficult to do. Now, what happened is, the HACCP plan has been established in 1996. But you look at, I just picked some examples from the FSIS website. In 1997, 12 million kilograms of the ground beef is recorded of E. coli 015CH7. In 2002, the beef trimmings and the ground beef has 9 million kilograms is recorded of E. coli 015CH7, and in 2007 have more happens, which is mechanical tenderized injected beefs. So what this tells us, it's not getting better. Now here I want to point out one thing. What I gave you the slides here is talk about recalls. It's not really the outbreaks yet. What means a recall? Which means the company find out the possibility of contaminated with E. coli 0157H7, they call the markets and trace them back. That's called a recall. There is a 3 different levels of the recall. Number 1 is severely affected human health. That is certainly is pathogen. Number 2 is allergy. So let's say you should not put egg and nuts there. Some people mistakenly mixed with egg or nuts. That will be allergic, which is allergy. The second level of the recall. The last level of the recall is, third level of the recall is mislabeling. So some products, the label is wrong, or you mislabeled, then the product has to be recalled. That's called the third level. Okay? Not only for that. This is what happened in 2012, about 16 years ago, 15 years ago. The FSIS had a new regulation, not only for 0157H7, the other serological typing, including 026, 45, 103, 111, 121, and 145. We call this, the nickname is Big 6. We find those Big 6 non-0157 E. coli had the same risk as 0157H7 because they all generate Shigella toxin. So Big 6, we call it non-0157 STEC. That represents Shigella toxin generate E. coli. Now obviously this is just bracket. So that's what we call it STEC. That is after 2012. So the regulation right now regarding non-intact beef products is E. coli 0157H7 and non-0157 Big 6. Both are zero tolerance. You cannot find one cell there. If you find one cell there, your product is adulterant. What you can do is well cooked to hot dog, which means become a ready to eat meat. You cannot sell as a raw product. Okay? So that's the E. coli, what we want to talk. Now, something else we want to mention about the E. coli. I mentioned it earlier. When we talk about the E. coli, they are different types. We have EHEC. That's called enterohemologic E. coli. We have ETEC, enterotoxigenic E. coli. We also have EIEC. That is called entero-invasive E. coli. And EPEC, enteropathogenic E. coli. What we are talking about here, the 0157H7 and non-0157H7, their fit category is enterohemological E. coli. They all will cause hemorrhagic uremic syndrome. Okay, just want to let you know, the symptoms are the same. Couple of years ago, about like 5 to 10 years ago, the USDA have a big multi-million grants to testing non-0157H7 and 0157H7. Lots of studies to do the comparison, but there is no results showing there is any difference. From my point, is both of these are similar gram-negative bacteria. Their cell wall structure are exactly the same. The only difference is the serological typing. So we call its attachment is different because flagella or those things will really attach to the cells, but the material inside are basically all the same. This regulation comes out all the research universities and the companies, they are developing the rapid testing for E. coli 0157H7. So by the time, there is a joke called USA, Universal Stack Analysis. And in the whole country is doing that. Lots of the rapid testing kits are developed by the time because it's a toxication issue. Okay, so that's all I have for E. coli 0157H7. And now 0157 should get toxin general E. coli. Next one is Listeria monocytogenes. We mention all the time. This pathogen originally from the soil. Nobody really cares. It's until 1998 to 2002. And you can look at what I wrote there. It involved in ready-to-eat meat products outbreaks. This is what we call is RTE, ready-to-eat meat products. The products involved or associated with Listeria monocytogenes including like hot dog, frankfurters, turkey breast, and hams. Those products, relatively speaking, it is high salt, high fat, and packaged, vacuum packaged even. And then they usually storage at the refrigerator temperature. But they still gonna cause the problem. The reason is Listeria monocytogenes can grow in the refrigerator temperature and can grow very well. So let's talk about that a little bit. Listeria species basically have these 6. Monocytogenes, Enochia, Silageri, Ivanori, Wilshmeri, and Greeny. We don't really care about the below 4. Those are just, we can say, normal Listeria species from the soil. We care about the top 1 we just mentioned. It's a pathogen, Listeria enochia. We sometimes worry about that because it is a surrogate bacteria for monocytogenes. So when the surrogate bacteria are using is the places you are not biosafety level 2 certified. You cannot use foodborne pathogen. You need to use some of the replacements. That is called surrogate bacteria. And most likely the surrogate bacteria has to be resistant to the intervention than the target pathogen. So I give you an example. Let's say if you are heating 60 degrees Celsius, you can kill 2 log of Listeria monocytogenes. Then how do you know enochia is a good surrogate? Listeria enochia has to be killing only 1.5 log, which means it's more resistant to the heat than Listeria monocytogenes. So this is something I want to mention. Now, monocytogenes, it is a very smart pathogen. And you can see all the characteristics here. What are they? They are gram-positive, non-endospore forming, facultative, which means they grow in the presence and absence of the oxygen. They grow better with oxygen. And they have a very strong moving capability because it is petritrichous. You look at the picture. Same as Proteus vulgaris, you can go everywhere. And you can see below, they can grow up to 10% of the salt. We mentioned about that. Sodium chloride is a natural preservative for bacterial control in our meat processing, although it's not very obvious. Hot dog and ham, they sometimes contain 10% of the salt. They can grow. And even this minimum amount of the salt can kill other bacteria, but let Listeria monocytogenes to dominant. And the pH growth is a wide range, from acid 4.1 to 9.6. That's alkaline environments. They can grow very low temperature, about 0.5 to 3 degrees Celsius. What is called? Do you remember this? Sacrophiles. Above that called sacrotrophs. Below that, what is the E. coli we call it? Mesophiles. Is that right? What is bacillus we call it? Thermophiles. What is geobacillus we call it? Hyperthermophiles. That's what I said. When you move to exam 4, luckily the exam 4 is not accumulated. Okay, it's only last section. You need to know. Don't forget about that. That's a bacteria can grow in the refrigerator temperature. It causes a problem. Another thing you also need to know. Look at the last one. Listeria monocytogenes can grow at very low water activity. That is very low for lots of the bacteria, for lots of the products. It's pretty much like a summer sausage and the salami. Usually those lactic acid fermented products is really dry, and even beef jerky. So they can grow very well, and it's also resistant to the salt. What terminology called resistant to salt? That's called halophiles, remember? Okay, so that's the characteristics. Listeria monocytogenes can grow very well. And we have 13 serotypes, but lots of that we don't really care. 95% is for those 3 species. And the cereval 4B accounts for 65 to 80% of the Listeria monocytogenes in the North America, which means basically Canada and Mexico and the United States. What's the toxication it will give to us? This is also a little bit old survey. Published at CDC website. In 2011, Listeria monocytogenes is estimated approximately 1,600 foodborne illness, 1,500 hospitalization, 260 deaths in the United States annually. So, FSIS have 3 alternatives. Number 1, post-lacerative treatment using antimicrobials. Number 2, post-lacerative treatment or antimicrobials. Okay, what this means? Post-lacerative, which means the hot dog has to be well cooked. That means it's ready to eat meat. What means antimicrobials? This is in the lab. I show you the bag. You see sodium potassium lactate, sodium diacetate. Those are antimicrobials. So you can, the one of the things you should do is well cook and using antimicrobials. Now, how are you going to use the antimicrobials? You could add the ingredients, which we call the emulsifying process. You could be spraying on the surface using a sprayer, or you could be dipping into the solution. So basically, there are 3 methods. Now, sometimes you say, you are post-lacerative. I don't want to use antimicrobials. Or sometimes, let's say I use antimicrobials. We can also use all. I'll give you a choice. But the last is, I don't want to do anything. I don't want to do any of that. You can, especially organic processing, that rely on sanitation, and microbial testing. Microbial sampling testing. rely on SSOP, Sanitation Standard Operation Procedures. And the Sanitation Standard Operation Procedure is unique for every processing, but it's more general. That has to be done before hazard plan setup, which means it's a prerequisite program. If you say, I don't want to do anything, then your SSS plan, SSOP has to be certified. And your sample has to do microbial testing. The standards from the microbial testing is not zero tolerance. It's less than 1 cell per 25 gram. And the testing, which is related to statistics results, we have class 1 and class 2. I used to teach, I don't talk about that anymore. It's related to the statistics. But there is one challenge thing here is that you should always know microbial sampling testing does not mean food safety. Because the food products have their very unique morphology. You're not going to test in every corner. There are a lot of the niche area you cannot touch. So think about when we're processing a poultry broil carcasses and we're using the shaking methods. But do you really make sure every niche area, all the skin surfaces has been sampled? Probably not. So microbial sampling testing does not equal 100% of the food safety. Also, related to the testing limitation. And also related to, we already talked about, it's viable but non-culturable, which means we may not can test that, but they're still viable. And sometimes we see the pathogenic gene there, but it's not really active at all. Now, related to the microbial testing, for E. coli, how we do that? Lots of the time, people get a beef. Put in the solution, what we did in the lab, usually with the special media or with tropic soil broth. Then we enriched at 35 degrees Celsius for only 9 hours, or 42 degrees Celsius for 9 hours. Then we go to run the PCR. We do a rapid testing to see Shigella toxin 1 and Shigella toxin 2. Because that is indication you have the toxin, you have the presence of the pathogen. Because this is we call intoxication and infection. Remember, we talked about the difference between that? That's called the infection. The toxin is there, indicates the presence of the pathogen, and it's liable. So, that's something about microbial testing. Listeria monocytogenes is a multiplex PCR. Different bands from the PCR images will indicate you what type of the species you have. So, just want to let you know, United States do have the regulation for Listeria monocytogenes control in the meat products, especially the ready-to-eat meat. Okay, we want to talk something else. In the history, we care much about food safety in the meat because we have the outbreaks. In the long term, we always think fresh produce is safe. And we eat a lot with your salad dressing. We do that all the time. The only thing people really care about fresh produce outbreaks is this guy. It's in 2006, in California, in Central Valley of the California. So, you're in West Virginia. You may not be familiar with California. That's San Francisco. Okay, that's Los Angeles. That's San Diego. That's Sacramento. That's what UC Davis is there. This is the Central Valley. This small area is responsible for 95% fresh produce in the United States, with 3 major companies Doe, Taylor Farm, and Fresh Express. If you go to buy a salad bag, go Kruger, more or less you will find the bag labeled as Doe, Taylor Farm, and the Fresh Express, or some of the local companies. Maybe in West Virginia you have some local companies, but that will happen. In this Central Valley, this is a huge fresh produce area. I went there 15 years ago to some of the pilot plant trial when I worked at ARS, USDA ARS. So originally people always think it's safe until this thing happens. That is a bagged spinach. People find isolates of E. coli 0157H7 and spread the whole United States. Result, almost 200 people. 26 states, 51% hospitalization. 71% of the female, 11% of the children under 5 years, that caused 3 deaths. There even got 1 or 2 cases in West Virginia. Lots of them in Pennsylvania and Ohio. See, the Ohio is a deep red. So they go there, which is widely spread. This is the reason the microbial safety in the fresh produce started to become a research topic. So literally about 20 years ago. So what we need to do, this comes out with a new regulation, not really new compared to HACCP, it's relatively new, called the FSMA, Food Safety Modernization Act. This is signed by President Barack Obama in January 4th, 2011. It's a federal register, it's a law. There are 7 major rules there. We're not going to worry and cover everything. For our food safety area, we are more concerned is produce safety. So they have a special documents for produce safety. From growing, harvesting, packing, hold on, and also human consumption. And more important we care about is preventive control. The preventive control here is pretty much is HACCP. So, in the long term, there is a joke by the time people, they just copy and paste it. And it's a fraudulent happens, quotation mark. Because lots of the regulations are very similar to the HACCP. But the major thing here is the preventive control of human food. They added allergy at the same risk level as foodborne pathogen. And they include the food products has to be well labeled. If you have fish, egg, nuts, shrimp, all types of the seafoods, they're all in the category which is allergy. So preventive control basically is the HACCP plus allergy control. We have a special certification program for the produce safety and also preventive controls. The produce safety, we did a very good training in West Virginia. We call it a trainer certification program. And the preventive controls, the best place is Pennsylvania State University. They did a really good for that. Some of you in the future, if you want to working in the food industry about the pathogen control, it's good to, during your amateur time, or summer or winter, to get those certification, which is very beneficial to your career to find a job. So that's something in addition to these regulations. Now what happened? It's very interesting is Listeria monocytogenes not only on the ready-to-eat meat, it also happened on the fresh produce, which is cantaloupe. This is Jason Farms in 2011 in Colorado, which has happened a multi-state outbreak. So what happened is, it's very interesting, the federal regulation and the FDA, the personnel went there to see their record keeping and documents. They find the Jason Farms about 3 days before the outbreak, their SSOP is ranked as excellent. But then 3 days later, the outbreaks happened. Now, cantaloupe is relatively very tough. If you know cantaloupe, that's what their surface looks like. And this is a niche area. Those type of the unsmoothed surface is very easy to harboring or cultivation or growing bacteria. It's looking like a net. So what type of the things they will have is biofilms. I mentioned earlier as a matrix of the bacteria. If you have the biofilm on the cantaloupe surfaces, it's resistant or 10 times, 100 times resistant to the antimicrobials compared to the vegetative cells, which means the cells in our lab. That's why it causes major outbreaks. It's about 147 infected in 28 states and 33 people died. And very ironically, this has happened in the summer 2011. This is what happened the FISMA just had is January 1st, 2011. It's very ironic. The new federal regulation of the food safety signed into the law just half a year later, you have another multi-state outbreaks. So once this one happens, it keeps the university extension specialists are very busy. They are do all the type of the training to digest about 500 pages of the FISMA. And I saw the produce safety regulation. The training book is like that. Very heavy, pretty much like a biochemistry textbook, like that heavy. So, because the extension specialists have to do the preparation by themselves, transfer those scientific language, become a layman language, and let the producers to learn what they are really talking about. So that's become a hot topic by like 15 years ago. Very quick, can show you some of our research. That's what I did. Collaborate with faculty in Wayne State University. You can see we get fresh produce from farmers markets. We find that around 109 samples, 3 of them are Listeria monocytogenes, 13 of them are Listeria inocia, lots of them are normal Listeria wilshmeri. And then we using PFGE, which is old genotyping. We group them into different colony complex. And what is interesting, you can see, lots of those coming from radish, romaine lettuce, potato, and romaine lettuce. And we also find that all of those samples tested the presence of Listeria monocytogenes. They also carry on the 5 genes, which is antibiotic resistant, which means those Listeria species not only by itself, it also carry on the genome is antibiotic resistant, which certainly means if you applying some antimicrobials may not be work very well, because for themselves it's already become antibiotic resistant. FISMA is a federal regulation, but they do give you some flexibility for the small or very small farmers. They gave 3-year average. If you sell less than 500,000, or your distribution your products within 275 miles, what does that say? If you're growing your produce at Charleston and you want to sell to Morgantown, you're waived for FISMA regulation. So this is very important for the state like West Virginia, because they relieve the legal burden from those small producers. And I don't think any of the producers here will have 3-year average sales is more than 500,000. That's huge. So what happened, at West Virginia, we do have a farmer's market, because those small producers go to the farmer's market. And you can see the farmer's market is very popular in the country. And the number is increased from only 17,000 in 1994. In 2019, right before the pandemic, it's around 80,700. This is what happens in Morgantown. And you can see there's two major farmers markets in Morgantown. And there's about 1535 vendors. They are selling Wednesday, Saturday. And the annual sales is approximately 0.5 million, and lots of these are produce. This is the research what we did. And you can see lots of those, we get the salmonella from tomato, pepper, cantaloupe, and spinach. The only thing we didn't find is the cucumber surfaces. It's more than what we find in the literature. Because most of the study in the literature, especially in California, they are less than 1% of salmonella in tomato and herbs. That's my very early study, about 8 years ago, when I just be here for 2 or 3 years. So, for local small producer or small farmer, what we should do, we maybe can flee away from FSMA, but we have to do something that is called a gap. That's not a commercial clause. It's a good agriculture practices. It's a quality assurance procedure from farm to the table. If before planting, during production, during harvesting and post-harvesting. Now, gap regulation is also very heavy. Not about 500 pages. I think it's about 100 pages. We have it. But what they are talking about? They talk about 4 items. Number 1 is safe soil. What means safe soil? Relate to the manure, animal manure, or plants manure sample. The manure cannot use a fresh one. Needs to be at least 48 hours older, because the fresh one will have generic E. coli and the coliform. Second, clean water. What means clean water? Zero tolerance of generic E. coli and the coliform. That's what we did in the lab. Clean surfaces. What means clean surfaces? A good sanitizing procedure, the cleaning and sanitizing procedure, to prevent the formation of the biofilms. What means clean hands? So wash your hands. Wash your hands, use antimicrobial soap for at least 25 to 30 seconds. How do you make sure that? You're not going to do the timing. Nobody going to do that. So, the way to do is you can sing a happy birthday song. When you finish singing, then you wash it. That's 25 seconds to 30 seconds. So that's the key points of the fundamental mentioned in the gap. Last, in the United States, we have the surveillance and the regulations for all of these. And lucky, regardless of the reorganization of the government agency by the current administration, these are still stay there, although they shrink a little bit. The first one called the CDC. The full name I already mentioned, the Centers for Disease Control and Prevention. What they do in the older time, they are using the methods very similar to what I did in the research. They do PFGE. So you want to have those patterns like that. They have a database. If you have an outbreak sample, you compare the database. If your bands compared to the database bands is less than 3 banding difference, we say 99% of match. So they have a PFGE pattern, puzzled geoelectrophoresis pattern. Recently, 3 years, they get rid of the PFGE. They are using whole genome sequencing. That's called HGS, which means a colony isolated on the agar plates. A single colony represents one genome name, one species name. They know all the ATGC, the sequencing, and they can compare their database, which passaging it is. This is called whole genome sequencing. Regarding the regulation, FDA and the USDA have a different category. USDA, we call, there is a special agency, FSIS, Food Safety Inspection Service. This agency is regulated for meat and poultry products. But you need to be careful. They also, for what? Processed egg. What is processed egg? If you have a hamburger, 2 bread, 1 piece of ham, 1 piece of cheese, you have bread, you have an egg right there, scrambled eggs there, that is FSIS regulated. How about the raw egg? The raw egg requires the salmonella control, that is for FDA. So for the egg products, it's a little bit tricky. Now FDA basically is for domestic and imported foods, and they have a new regulation which is called FSMA, we just mentioned. And the full name is Food and Drug Administration. I want to have these slides here is not only to let you know the regulation. I want to also tell you, in the summertime, originally, I don't know right now, CDC, FDA, and FSIS, they all have summer internship for undergraduates to work there, if you're interested about microbiology. And I tell you one thing, 3 years ago, when I walk in the parking lot, there's one of the students from plant science division that come here to shake my hands. They said, Dr. Shen, I like your class because I got the internship in the summer from CDC to do the 3 months training there for microbiology research. They all have the summer internship program, so you can look for it. I believe right now, the FSIS may need more people to do the microbial testing. Because if you look at the budget, everything else is cutting. They only put more budget at FSIS related to food safety with the microbial sampling testing. So that's the place you can look in for if you have some summer internship opportunity. That is the purpose of these slides. I want you to know the full name of these agencies, their functions, briefly, of course, but more important, these are the places you could find an internship job, or maybe in the future have a permanent job there. That's why I want to have these slides here to talk about this. Okay? So that's all I have today. After Thanksgiving holiday, we're going to talk about the poultry meat processing.